So far we've talked about mass spectrometry-based proteomics, which is a large part of proteomics, but there's definitely other ways to do proteomics. And one big area is to look at protein interactions with microscopy. And so today we're going to continue the signal processing that we started a few lectures ago, and where we looked at one-dimensional signals. And now we're going to move to two-dimensional signals, which is images, and how to process those. Because probably in a lot of projects that you will be involved in, you'll be exposed to images, and you'll generate your own images. And it can often now, I mean, the people I speak to, they look at their images and analyze them that way. But if you want to do a little bit more high throughput, collect more images to do statistics, then it's useful to do some automation of the and it's actually quite straightforward to do that. And so programming languages like Python and a lot of other languages have a lot of modules that you can use. So you can actually, with not too much effort, do the types of analysis that I'm going to go through. But just first, a little bit of recap on the first signal processing lecture, where we looked at one-dimensional signals. So you probably remember that we talked about Fourier transform. So this is a transformation between frequency space and the linear space. And then you can go back and forth, and it can be useful to transform it into frequency space, filter in frequency space, and then transform it back. Then we talked about what is a signal and how we can detect and characterize signals, and also how we characterize noise, and a few different ways how we can model signal, both signal and noise. We also talked about how we can use, if we have a model for signal noise, how we can then do simulations to select thresholds and different parameters for how to distinguish between signal and noise. We also talked a lot about, quite a lot about filters. So one example was a low-pass filter, where we remove high frequencies to smooth the signal, or we can also do high-pass filter to which the example that we did was called adaptive background correction, to separate signals that are from peaks that are very close to each other. So then we talked about different detection methods, like that we can, based on either moving average and root mean square deviation. And so the examples that we talked about here were on purpose quite simple methods where we can really understand easily what the method, how they work. And so people have worked a lot on signal processing and developed a lot of different methods that we didn't touch upon. But I think by looking at these simple methods, we covered a lot of the space of what can be done then also, but with maybe more advanced methods. Then we talked about convolution, which is how we can take a signal and convolute it with another signal, and this corresponds to if we have if we have a time invariant signal system and give it an input signal, and if it has a linear response, that would be, we can mathematically describe that as a convolution. Then we also looked at similarity between different signals, and the main way we did was cross-correlation. Then we looked at autocorrelation, where we can correlate the signal with itself to look for periodicities. We talked about the dot product, which is also a measure how similar two signals are, and that can be used in, for example, for if we do spectral library searching. We talked a little bit about coincidence, that which is a very powerful method if one can measure sort of do orthogonal measurements of the same underlying thing. So, for example, in mass spectrometry, the example would be to measure both the precursor ion and its fragments. So each fragment would be sort of a different measurement that of the underlying quantity, and it would be probably have different kinds of noise. So measuring those together would be a coincidence measurement where we can really enhance the signal and suppress the noise. And we talked a little bit about how one can quantify a peak by either looking at its height or area. And we also talked about sampling, how often one needs to sample a peak to get a good estimate of its area. So that sort of summarizes. So just one highlight was when we looked at peak finding, and we discussed a method to do this, and the steps were to first start with characterizing the signal and the noise, then make a model of the data, and the third step would be select the detection method, and then finally do simulation on this model with the detection method, and to select the thresholds to use for peak detection. So now if we move on to 2 dimensions, to images, we're going to start by actually making a slice through the image so that we get a one-dimensional signal that we're used to, and looking at that first. But then we're going to go on and look at some of the methods that are made for image processing that takes into account the correlation in both dimensions and not just in one dimension. And then we're going to talk about a little bit how to set thresholds in 2 dimensions, how to find and characterize objects, and then when we have found objects, then to classify them according to their shape, and then finally we're going to, if we see an image, we can use simulation to test if the observations we see are random. So these are the kinds of things that we'll do. So we'll start with looking at how an image is represented in Python. And it's quite simple. It's a 2-dimensional numerical array. So we can we'll have the rows of the array are along the x-axis and the columns along the y-axis, and the origin is up in the top left corner. So then there are several different packages that we can use. So within SciPy there is an image processing tool. There is a scikit-image, Mahotas, Matplotlib, that we can, and these contains a lot of the functions that we're going to go through that can operate on images. So just a very simple example it would be to that we get here 12 gel images, which each gel image has a lane with molecular markers, and then a few more lanes, in this case a total of 8 lanes in each gel. And now what we want to do is to organize this and find, first of all, cut them up, this picture up, so that each is on its own. Then we want to straighten it out a little bit, and then we want to quantify, and we want to also make that the mass scale is the same using this ladder. So now if we put them just in one row here, and then look at the red and the green signal, we see as we go through from left to right, we see peaks with in the red signal. We see smaller peaks in the green. So, but since we know that in our case these ladders are the ones that usually are going to give the highest red peaks, and those are the ones we want to find to be able to say where a new gel starts, so we can cut them up. So in this case we see all sorts of peaks, but sometimes there is quite a lot of red signal, like in this case in the middle of a gel, and we want to then, so the first step here, we can detect the peaks in the red signal here as we go through the image, and then take each of these and look in the other direction what it looks like. So this is, for example, one example where we look along a lane, and then we can decide whether this is a proper. So the red signal is what we measure first, and then the gray signal is a background that we set, and then we subtract that and then get the black signal, which are should be the cleaned up bands. And then what we can do is to look at the relative distance between these. So we should see that in this case we can recognize it. So there is, for example, a much bigger space between these two bands than between these two bands. So these are the things that then we can use. And if we look at more, so these are 3 of the bands that of the marker lanes, and sometimes it works really well. This one, although this one is a little weak here, but in this case we have a nice signal, but in this case this one is very weak. So the things like that happen all the time, and we need to be able to deal with that. And then the ones that we want to not detect are these. And you see that the relative distances between the lanes here in this lower panel is very different. So we can then set a criteria for which is a marker lane by the number of strong red bands. We can have the intensity variation, although we have to take into account that sometimes for some unexplained reason this one is very weak here. We should have a low green signal throughout these. I guess actually in these 3 lanes we can't detect the green signal at all, and then the relative band locations. And if we do this, we can pretty easily automatically detect the lanes. So now the next step is to straighten the lanes. So because we have these, you see this lane, it's been quite distorted. So what we can do there is to just try to find a minima by both moving these this line along the x-axis and also looking at different angles to find minima between the lanes. And then we can straighten it out. So you see here this band is quite crooked, but here it's much straighter. It's still not perfect, but it still now fits within this raster very well. So and then the next step is then the calibration. So then what we do is we go back to the marker lane, but now not just decide whether it is a marker lane or not, but then detect which bands correspond to which mass, and then put those, so straight bend, stretch the gel so that all of them are the same on the same scale. And then we can separate the red signal from the green signal, for example, if that's what we want, and annotate this with different arrows and so on. And then the next step is, after all this is done, that we want to measure the intensity of the different bands. Definitely these steps are quite straight, and you can do all this in Python quite well. In this case, you probably have to add the extra code for that's specific to the gel, but you can use a lot of packages, so the code becomes quite short. So another example is from a project. It's a collaboration with Paolo Mita and Jeff Bocke, this example. And here they are assessing the quality of antibodies. So they bind antibodies onto beads and then see how much of their fluorescent protein binds. So what you have these, you see these rims here. So what happens here is the signal so these beads are sort of semi-transparent, and you get your antibodies binding on the surface. So when you look on an image in a two-dimensional projection, you're going to have much more signal on the rim than in the middle. And you can see why that is. If you have a sphere that has it on the side, there will be a much longer path through. And in this case, the noise seems to be that there is some bulk fluorescence from the beads. And this is now a titration, so this is more and more material, and you see a clearer and clearer image. But even for the low amounts, you see this rim, and then for the background, you see that they're round balls with the bulk fluorescence, and sometimes quite a lot of fluorescence for some reason. What we want, the goal with this was to analyze this in a way that the background should be 0 for all the controls, and for the experiment it should be linear with the amount, and one should be able to, of course, distinguish as low amounts as possible from background as possible. The simplest way to do this, so here we have what we want to achieve. The simplest way, we can just take the average pixel intensity in the image. This is very easy to do. Then we get a high background, but we do get a linear signal. But the problem is that we have this high background. But so then what we can do is to look at the what so this is the distribution of pixel intensities, both in the experiment in red here and the control. So first of all, what we see is that we have in the experiment the distribution goes to much higher values, and that's we can see from the image here also. But we see that there is not a very clear separation between what's the background and what's the experiment in these distributions, because there is quite a bit of overlap. And then if we look at this from for the other amounts, we see that as we go down in amount, there is less and less separation, and we have much more of the signal is overlapping, and it's difficult to select from this pixel intensity distribution a good cutoff between intensity, between the experiment and the control. So the next step, what we can do is, and the other thing we observe is that the lowest intensity is not 0 in any of these. So another addition to the thing is we can take the average pixel intensity, but we can first subtract the minimum. And that actually improves. We have now, if we have the measure as the ratio here, so here we have now about a factor of 4 difference, and here we had about a factor of 2. So we're getting, it's getting better and better even with these very simple methods. But now, again, we can go back and create a one-dimensional signal for us to analyze. And then if we go through in the middle of this image through a slice, we see that we get these peaks, and when we cross a rim, we get these thin peaks that correspond to the rim. And then if we do the same for all of these, we see that the signal contains these thin peaks, even down to quite low amounts, and the background is where we see the shape of the balls instead, and no rim effect. We get these slowly moving. So we can recognize this from the one-dimensional, the homework that you did, where we had to find smoothing parameters to separate, so that remove the wide peak in the middle and sort of enhance these thin signals. And you remember that one could choose a background that followed the larger peak, but did not follow, was wide enough so that it didn't remove the smaller peaks. And then after another smoothing, we could get that the 3 thin peaks were enhanced. And this is exactly what we can do here. So we have these thin peaks that we want to enhance, and if we smooth it, we get a smoothing. We choose a smoothing that follows the wider peaks, but don't leave us the thin ones. So then, if we subtract, we can do this for all of them, and finally we get and subtract the background. We see that for the background case, we just see very little noise, but for the signal we're left with these thin peaks. So now if we use this, now we just integrate these, we get some quite good. Now we have a factor of about 10 here, so we're getting closer and closer. And then if we set a small threshold of maybe 3%, we get very close to the situation where we want to be. And so this just recaps everything. So the goal was 0 background and a linear signal. And then first we tried just to integrate the whole image and have a high background. We subtract the minimum. Then we get a slightly lower background. If we then slice it in one dimension and go through, do every slice, then we can get very close to where we want to be. Just a very low background here. So now now we haven't really done proper image analysis because we haven't taken into account the correlation in both dimensions. We just looked in one dimension. So in summary, one can get often pretty far, at least with these kinds of analysis that I've shown so far, with just doing one-dimensional analysis. But of course, here we have it's two-dimensional. We should really take into account that these are circular shapes, and we should do the analysis properly. So the kinds of things that we can do is, so first we need to threshold. And the thresholding is that we decide what is background and what is foreground. So this is just one example of a picture where there is a photographer in the foreground, and then there are some buildings and sky and grass in the background. So if we look at the histogram of intensities, we then can decide, we set a threshold somewhere and decide what is foreground and background. So in this case, it's mainly the photographer that was selected, but some of the background, the buildings were also considered foreground based on this simple threshold. And then the question is, how does one select this threshold? And there are different suggestions, but one, it's called Otsu's method, is that you select this threshold so that the variance in the two classes is minimized. And this is a usually a rational way to select it. It's not necessarily always the best way to select it, but it's so there are have been proposed many other ways also. So now if we look at our original image, we're going to go through a few different analysis steps. So one is we can set a constant threshold and then say that that's globally over the whole image, and decide what's foreground and background. So then, I mean, you can see here that we, for example, if we do this, we lose something. So this, if you look at, as an example, at this bead here, we didn't detect, just partially detected some parts of it. It's even worse, like this bead here. It's not very clear it's clear on my screen, but at least the thresholding did not find it at all. So the constant threshold doesn't work really well with this image. So what we can do is do an adaptive threshold that will take into account the local environment. And then we can find so this object that we barely see here, it was able to detect in this case. So so especially for images that like this, an adaptive thresholding is much better. But now, as you see, it finds a lot of other things. Finds a lot of small features inside of each ball that we don't really want. So then what we can do is add on other steps to the analysis. And one is called erosion, where we remove the smaller objects. So now we get a much cleaner picture where each ring is much cleaner inside. It doesn't always work, but in general it works quite well. So then now the other thing we can do is that we can focus in on just the objects that are continuous and that are larger. So in this case, if we have removed smaller objects. And the nice thing with, as I mentioned before, these libraries that we have in Python and other programming language, high-level programming languages, it's just each of these steps is pretty much one line of code that you can. So it's really straightforward. Now we can fill the objects if we want to do that. We can then segment them so that we separate each object to itself. And watershed segmentation is the most widely used segmentation. So the way it works is that one inverts the image, and what a positive, a peak becomes a minimum. And then what one starts filling this, imagining filling it is with water, and then the watershed line is where different minima meet. And those are the ones that this algorithm detects. So you take minima and then find the watershed line in between here. So that one goes from this to something that looks like that. We have, in this case, these three, there were 4 objects that were separate, but then in this case it also succeeded to separate these objects that were partially overlapping. So now we can move to another example where we're going to look at overlap between objects. So this is super resolution microscopy, looking at 2 different proteins. And the idea is to see how often they overlap. So this is what you see here is one nucleus, and then these 2 proteins throughout the nucleus. And so what we can do here, so this is a much easier image. Here we can actually use a global threshold. So what we do is we start with just separating the red and the green image, and set a constant threshold. So this is the original image here, and then this is the thresholded image. So if we compare it, you see here in the original image we have a grayscale, but in the thresholded image we decide by a constant threshold what is background and what is an object. And we set it so we only have, it becomes binary, it's either white or black. So just here we have the thresholded image, which we have either 0 is black in our case and white would be 1. And then we go from here to labeling the particles. Yeah?
Do we always have to convert image into binary position, like white or black?
No. I mean, we don't have to. In this case, it makes sense to do that because we want to look at overlaps. But you could also analyze, I mean, if you want, like we did for the one-dimensional image, we integrate under a peak. And that we can do here in 2 dimensions also.
Okay, at that point, we cannot use some kind of array. Can we use it?
Sorry, some kind of...
Arrays we have used for the...
Yeah, no. So we can keep... So the original image is that at each pixel we have many different values. So that we have... So it's represented as an array. And we can then do the integration in 2 dimensions. But we still have to decide what's the edge of the object some way. But for this particular purpose, it was actually easier to threshold and decide that it's either part of the object or not part of the object. And then then one would go through this and label. And the idea is that find each sort of area, each object, and then get its properties. So we have the original image, and then we get a list of objects that we find. And then again, sorry, this doesn't show up very well on the screen. But there are small areas around the objects. Oh, this is a little bit better, zooming in. So then, for each of these objects, then we can measure their area, where they're located, how circular they are. Maybe we can just measure the major and minor axis length, their intensity, either the minimum, mean, or maximum, we can then use all these things to we can extract all these things automatically from the image and for example create histograms that will describe how these things change when we do some manipulation to the cells. The other thing we can look at is overlap between particles. So for example, here we have 2 green particles that are on their own, but then we have 1 green particle that overlaps with a red particle. So then we can look at how also these overlaps can be treated as a third particle, of course. And then when we look at overlapping particles, we can also classify them into different kinds of structures. For example, this one, the magenta particle sits almost at the end of the green fiber. Here it sits right at the end, and here it sits in the middle. So if we want to automatically classify our overlapping particles, one thing we can do is, for example, find the green fibers and then take a slice through them and see where the magenta is distributed and where the yellow here is the overlap between the two. So in this case, we see that the magenta is somewhere in the middle of the green, but in the other case we see that it's at the end. So here it overlaps, but it extends beyond it. So by going to by first doing the two-dimensional image analysis, but then switching over to looking at one dimension, we can it's a very powerful way to then classify the structures into these categories. So then the other question is about randomness. We see this image, and one thing we can ask ourselves, are these overlaps just random? If we would put at random this many particles in a box, would they have the overlaps that we measure? That's, of course, a very basic question that one should always ask. Now, it doesn't look like it's random, but it's always best to test it. So the one thing one can do is Monte Carlo simulations. So here we take the properties. So we take the number of, the density of the green particles, density of the magenta particles, their size and shape distributions, and then just randomly lay them down, and then see how much overlap one gets. And this is something I would recommend doing when if one looks at images and sees some kind of structure. It's always good to test if it just could happen at random. The other thing that we have with this interaction, that it's possible to do more colors. This is now 3 colors. And you can imagine with 2 colors we could have overlaps. Now with 3 colors we can have even more different either overlaps between all the 3 or overlaps between the pairs. And so there is a lot of different information we can extract. The other thing is that it's actually possible to do more than 3, probably 4 or 6 colors even. So that's, of course, adds a lot to the complexity, but also adds a lot to the possibilities that one can do. And then we can also have more dimensions. So far we've looked at 2 dimension, but sometimes it's possible to look at it in 3 dimension. So this is one example of if we now turn this around, you see that it's looking at it in 3 dimension becomes even more interesting. So things that look like when you look at the 2-dimensional projection, might overlap, in 3 dimensions they might not. So that's another complexity that we can add to our analysis, but again, giving rise to more information. So this is, this is what I wanted to tell you today about. So this is about all about looking at images. And in our case, especially microscopy images of protein-protein interactions, which probably will be the main application. And well, we talked about the few different methods to apply where one can analyze. And the take-home message is that if you ever generate your own images in the lab, it's really not that difficult to write your own code in Python to analyze those automatically. And that's definitely preferable. Of course, you will always start out by looking at it and observing things, but then to test your hypothesis on a larger set of images, it's definitely much better to do it with some code that can do it automatically. So that's would be my recommendation. So that was everything I had for